reactive oxygen species formation in diseases that we are very much focused on cardiac disease, but I'm also going to show you how this concept that we have been developing in the last more than couple of years can actually apply also to diseases affecting skeletal muscle as well. So I'm going to start by saying some very obvious things that you probably know better than me on how mitochondria are important for myocytes. Here I am showing you an EM that is relative to the mitochondrial organization and abundance in the heart, but in skeletal, in muscle myocytes, this is quite similar. So the main role of mitochondria in a myocyte is to produce ATP through oxidative phosphorylation to sustain the very high energy demand imposed by the beating heart. So the role is to produce ATP that would sustain then contraction and relaxation. Mitochondria are very abundant in myocytes, and they have this very peculiar spatial organization. So they are organized in these tightly packed rows of mitochondria between the myofibrils and also beneath the sarcolemma. And this spatial organization and abundance actually is key for a very efficient and localized ATP delivery that is required to sustain contraction, metabolism, and ion homeostasis. Mitochondria can also act as cytosolic calcium buffers. In fact, in the heart, mitochondria uptake cytosolic calcium on a beat-to-beat basis, and they also serve a signaling role through the production of metabolites or even reactive oxygen species that can then basically transmit the signal and message to other parts of the cell. Reactive oxygen species are certainly important signaling molecules, but the imbalance between ROS formation and elimination has been associated with the development of a number of not only cardiac, but overall in general with a number of pathologies. Here I'm listing only some of the pathologies affecting the heart in which ROS play a major role. And it's important to know that in order to target ROS formation, we need to know where these reactive oxygen species are being formed. Here is a list of only some of the sites within the cell where ROS can be generated, and these sites are important for pathogenesis of several diseases, for instance, NADPH oxidase, xanthine oxidase, uncoupled NOS, and obviously mitochondria, which are considered to be a major source of site for ROS formation in the cell. However, within the mitochondria, there are also several sites where ROS can be generated, and probably the best described site is the mitochondrial respiratory chain. So we all know very well that at the level of complexes 1 and 3, electrons can escape the respiratory chain and partially reduce oxygen to superoxide. Then we have also P66-Chic, which is a cytosolic adapter protein that upon stress can translocate to mitochondria and result in H2O2 formation. And then there are monoamino oxidases, flavoenzymes localized at the outer mitochondrial membrane that can also produce H2O2. So monoamino oxidases caught our attention because of this paper that established that in brain isolated mitochondria, the steady state H2O2 formation that is obtained during the monoamino oxidase catalyzed oxidative deamination of its substrate tyramine is 48 times higher than the one that is generating during the oxidation of substrates through complex 2 and in presence of antimycin A. And to note that this is a condition in which the respiratory chain is generating the maximal amount of ROS. So to briefly introduce monoamino oxidases, they exist in two isoforms, A and B. As I mentioned, they are localized at the outer mitochondrial membrane, and they catalyze this oxidative deamination of catecholamines and neurotransmitters. During this process, they generate aldehydes, ammonia, and H2O2. And there are inhibitors available also in the clinic for monoamino oxidases because they have been extensively studied at the level of the central nervous system in relation to their capacity to regulate levels of their substrates, specifically serotonin or catecholamines and other neurotransmitters that are important in several psychiatric or neurological disorders. However, the role of MAOs has been studied very little in peripheral tissues, for instance, in the heart, and especially very little attention has been given to the products of its activity, for instance, hydrogen peroxide. So the questions that we've asked is whether MAO activity is important in the heart, and whether products of MAO activity can also have a role, for instance, in pathological situations, whether their excessive formation can be also damaging. So before going into tackling these questions, I would just like to illustrate how this system works in physiological conditions, presumably. So at the interface between the sympathetic nerve efferents, and in this case, I'm representing a cardiac myocyte here, but it can be any other cell. When catecholamines that are stored in the neuronal vesicles, when they are released, they interact with the receptors localized at the sarcolemma of cardiac myocytes. And once this interaction is over, they are rapidly reuptaken through the specific dedicated transporters, and they are recycled back into the vesicles in the nerve terminals. This happens with 92% of the neurotransmitter that has been released. Only 4% escapes into the circulation, and another 4% can be uptaken by the extraneuronal cell through the extraneuronal monoamine transporter. And once in the cytoplasm, it is catalyzed by MAOs, resulting in the formation of H2O2 that in physiological conditions is presumably rapidly and efficiently neutralized by the cellular antioxidant systems. So to see whether by pushing this system, we can observe some deleterious changes, we took neonatal cardiac myocytes and we used this genetically encoded H2O2 sensitive probe called HYPER that is very convenient because it can be targeted. For instance, here we target it to mitochondria or to the cytosol. So if we give these cells transfected either with one of the two sensors, if we give them MAO substrate dopamine, we see that within the mitochondria, H2O2 formation occurs rapidly and it's quite sustained, whereas in the cytosol, it is much delayed and it occurs to lower levels. So if we quantify this, we see that within the mitochondria, we see quite a dramatic H2O2 formation if we stimulate MAO activity. And importantly, if we give a pharmacological inhibitor of the enzyme, in this case, pargilin, we can completely block this mitochondrial ROS formation. So we know, I already said this, and I just showed you that mitochondria may be a ROS source. However, it is very well known also that mitochondria can be a ROS target. In fact, there are several components within the mitochondria that can be targeted and damaged by oxidative changes. For instance, mitochondrial DNA, several complexes of the mitochondrial respiratory chain can be impaired through oxidation, ATP synthase, several subunits of ATP synthase are also targets for oxidation, but importantly, this can also result in the opening of the permeability transition pore and then activation of pathways that lead to cell death and tissue dysfunction. So to see whether MAO-produced ROS can also target mitochondrial function, we went back to our cells, we feed them with dopamine, we looked at mitochondrial membrane potential by staining them with TMRM. However, we did not observe any changes in TMRM fluorescence intensity. So to see whether there is a latent dysfunction and whether mitochondria are somehow compensating for an impairment that is probably latent, we used oligomycin. And this is just to briefly explain how oligomycin works. So in normal conditions, the transport of the electrons across the respiratory chain is coupled to the pumping of the protons across complexes 1, 3, and 4. This generates this proton gradient that is then used at the level of the ATP synthase to drive ATP synthesis. So in case there is a problem at the level of the respiratory chain, in order to maintain membrane potential, ATP synthase can start working in reverse by pumping the protons out of the matrix into the intermembrane space. And this is coupled to the hydrolysis of glycolytically synthesized ATP. So if we use oligomycin to block ATP synthase and block this mechanism of compensation, we would immediately see a drop in mitochondrial membrane potential. So to see whether this was the case, we took adult cardiac myocytes in this case, isolated from either wild type or MAOB knockout mice. If we give them only oligomycin, we in fact do not see any differences in TMRM fluorescence intensity. If we give oligomycin together with MAO substrate dopamine, after 20 minutes, we already see that there are single mitochondria that start to depolarize in the wild type mice. And after 45 minutes, this loss of mitochondrial membrane potential is complete, while in the MAOB knockout myocytes, this does not happen. And those mitochondria depolarize only after we administer FCCP. And this is quantified here. So these results suggest that MAO-dependent Ros formation within the mitochondria can actually target mitochondria to induce mitochondrial dysfunction. So in this case, we use an in vitro system and we push MAO activity by feeding the substrate. So to see whether any of this is relevant in vivo, one of the first models that we have used is the pressure overload-induced heart failure. Heart failure is known to be characterized by spillover of catecholamines into the plasma. So this is a condition in which we thought that actually MAO activity may be prominent also in the extraneuronal tissues. So initially we took MAOA neo mice that are act as dominant negative and do not have MAO activity. We subject either wild type or MAO neo mice to pressure overload by transverse aortic constriction for 9 weeks. And we then looked at cardiac function by measuring pressure volume loop relationships. And we see that in wild types hearts, the loops shift to the right. This is because of the increase of the left ventricle chamber, and this is measured here by end systolic and end diastolic volumes that are increased in the wild type mice, and ejection fraction and index of cardiac function is reduced. However, in the mice that lack MAO activity, this function and changes in LV dimensions did not occur upon pressure overload. We observed similar features also in the MAOB knockout mice subjected to pressure overload, and also in this case, there is an increase in chamber dimensions, so there is LV dilation in the wild type mice, but in the mice that lack MAOB activity, both cardiac structure and function are completely preserved under pressure overload. Another pathology that is very affecting quite a big number of people worldwide is diabetes. And diabetes and diabetic complications have a common denominator, which is oxidative stress and mitochondrial dysfunction. So we wanted to see whether this idea of MAO as a central player in mitochondrial dysfunction can also play a role in cells that are exposed to the diabetic milieu. So we took cardiac myocytes again, and we cultured them either with high glucose or a combination of high glucose and pro-inflammatory cytokine IL-1 beta. Since diabetes is characterized by a very strong inflammatory component, we wanted to mimic better the conditions in vitro. And what we did see is that myocytes the myocytes affected with this mitochondrially targeted hyper show an increased ROS formation, both either when they're cultured with only high glucose or a combination of high glucose and IL-1 beta. However, if we pharmacologically target MAO using pargilin, this increase does not occur. And we have also confirmed this by silencing MAO expression by approximately 80%, and we observed the same kind of protection. So to see whether this condition is associated also to an impairment in mitochondrial function, we took adult cardiac myocyte, we stained them with TMRM to look at mitochondrial membrane potential. And we did see again that either with high glucose or a combination of high glucose and IL-1 beta, we do see a drop in mitochondrial membrane potential that can be prevented by pharmacological MAO inhibition. So to see whether this was due to an impairment in mitochondrial respiration, we permeabilized these cells treated in these conditions, and we measured mitochondrial oxygen consumption. However, either in state 4 or state 3, but we did not see any differences among the groups. So we hypothesized, as I mentioned before, there are other targets also of Ros within the mitochondria, and one of them is that oxidative stress can induce permeability transition pore opening. So we hypothesized that this might be happening here. So we went back to the TMRM experiment where we culture cells with either high glucose or high glucose IL-1 beta. We see a drop in mitochondrial membrane potential. But if we culture cells also in presence of cyclosporin A, we see that this depolarization of mitochondria can be completely prevented. So this suggests that MAO-dependent Ros formation is upstream of the PTP opening and can induce the opening of the permeability transition pore to provoke and result in mitochondrial dysfunction. We wanted to also confirm whether any of this is important in vivo. So we injected mice with streptozotocin to induce hyperglycemia. We treat them either with vehicle or with the MAO inhibitor pargilin. And after 12 weeks, we measured cardiac function. Here we measured ejection fraction, and we did not observe any differences between the groups. And this is something to be expected because one of the first features of cardiac impairment in diabetic patients is actually diastolic dysfunction without an overt heart failure. So to see whether this was the case, we looked at diastolic stiffness, and in fact, it was increased in these diabetic mice. But this increase was significantly reduced in mice that were injected with pargilin. This was also accompanied by a reduction in the oxidative stress. We measured here 4-HNE as an index of lipid peroxidation, as well as also of ER stress, which is another factor that risk factor that contributes to the development of diabetic complications. In fact, we see that when we block MAO activity, markers of ER stress such as ATF4 or GAT34 are dramatically reduced, suggesting that an impairment in mitochondrial redox status and function is upstream of the ER stress, at least in these conditions. Another cardiac pathology that we have addressed very recently, this is a paper published this year, pertains to the cardiotoxicity induced by doxorubicin, which is a major problem in cancer patients that are receiving chemotherapy. So we've wanted to see whether MAO does play a role there, because mitochondria is central to this doxorubicin-induced cardiac toxicity. And in fact, we did see, here we measured the mitochondrial glutathione redox potential with this sensor targeted to mitochondria. And we do see that when we give doxorubicin, the ratio between oxidized and reduced glutathione is dramatically increased, and it can be prevented when cells are also treated with the MAO inhibitor pargilin. We also see an impairment in the calcium transients in the cytosol. For instance, frequency is significantly reduced, and SR calcium load appears to be reduced, but all of these changes can be prevented in the presence of the MAO inhibitor pargilin. And also in this case, we moved to in vivo. We injected mice with doxorubicin, and after 5 weeks, we looked at cardiac function. Here I'm showing you fractional shortening and ejection fraction that are significantly reduced in these doxorubicin-treated mice, but mice that received both doxorubicin and pargilin show preserved LV structure. This is end-systolic dimension and also function in terms of fractional shortening and ejection fraction. So just to summarize this cardiac part, I think I have shown that MAO activation is deleterious in several pathological conditions and affects the heart and contributes to left ventricle remodeling and dysfunction. And either genetic or pharmacological MAO inhibition prevents oxidative stress, preserves mitochondrial function, and it affords cardioprotection in several pathological conditions. I would just like to conclude by saying, showing you actually, that this concept is relevant also for skeletal muscle. So Fabio De Lisa's group several years ago has investigated whether this role of MAO as a central player in the mitochondrial Ros formation and mitochondrial dysfunction is associated to the pathogenesis of muscular dystrophy, which are also characterized by redox change, oxidative changes, and also mitochondrial dysfunction. So in that project, they have used collagen 6 knockout mice developed by Paolo Bonaldo's group. And they've seen that monoamino oxidase activity is upregulated in significantly higher in the muscle from these collagen knockout mice. And this increase in MAO activity is associated with an increased oxidative stress measured by DHE fluorescence here, both or either in gastrocnemius muscle, diaphragm, or tibialis anterior. So these are the muscle from this collagen 6 knockout mice. But if these mice are treated with the MAO inhibitor pargilin, so MAO activity is completely abolished, obviously, but this inhibition of MAO activity completely prevents the increase in oxidative stress that characterizes these muscles. Another target of reactive oxygen species in the cell that is very important for contractile impairment are contractile proteins. So among them, tropomyosin is very susceptible to oxidation. And this can be also visualized in Western blots by running the samples in non-reducing conditions. So here I'm showing non-reducing conditions in the absence of beta-mercaptoethanol. And since these oxidative changes are reversible, if you run the samples in reducing conditions, so in presence of beta-mercaptoethanol, you can basically abolish all these changes to confirm that they're reversible. So this is tropomyosin Western blot, and this is the tropomyosin monomer. Since it has a cysteine residue that can be oxidized, you can also, in case of oxidation, you can visualize disulfide cross-bridge formation between different molecules of tropomyosin, shown here, or other even higher molecular weight complexes that are most likely due to formation of the complexes between tropomyosin and other proteins. Importantly, the increase in this disulfide cross-bridge formation in tropomyosin is significantly increased in these collagen 6 knockout mice. And mice that were treated with MAO inhibitor pargilin show significantly lower disulfide cross-bridge formation, suggesting that MAO-dependent Ros can target sarcomeric proteins to induce their oxidation. Importantly, this is also relevant from a functional point of view. So if these collagen 6 knockout mice are put on a running wheel, they can run quite less compared to the wild type litter mates. Whereas mice that have been treated with pargilin actually show a completely preserved exercise capacity and can run significantly more compared to the collagen 6 knockout mice treated with vehicle. And basically their capacity to perform exercise is comparable to the wild type mice. These results have been also reproduced in another model of muscular dystrophy, which is the MDX mice, and basically the results are the same. And another feature of this muscular dystrophy phenotype is that it also affects the heart. And this is studied quite less. Obviously, we are heart people, so we are interested also in that. And I would just like to show you some very preliminary data that we have now been developing and pertain to the structural changes that occur in the left ventricle of this, in this case, I'm showing you MDX mice. So basically what has been shown is that patients with muscular dystrophy also suffer from cardiomyopathy at some point. And this has also been observed in animal models of muscular dystrophy, for instance, in the MDX mice. The thing is that cardiac changes are observed quite late, for instance, after at least after 12 months in mice. And while changes at the level of the muscle occur much earlier. And one of the things that has been observed in the MDX, in the muscular dystrophy patients, Duchenne, for instance, is that approximately 30% of the children present left ventricular non-compaction, which is shown by this deep ventricular intratabecular recesses, and that this is associated with the higher mortality in Duchenne muscular dystrophy patients. So this is very important, and it's something new. And it has never been observed in any of the animal models of muscular dystrophy. So we looked into this actually very young, 3 months old MDX mice, and we saw that MDX mice show actually even a bigger left ventricle chamber, but they show this very big trabeculation, important trabeculation and non-compaction of the left ventricle. This is thought to be a congenital disorder, so it's a congenital morphological disorder, but in in Duchenne patients, it has been shown to occur also, that it can also be acquired. The mechanisms underlying this acquired form of non-compaction are not clear, so we are now trying to elucidate whether mitochondrial Ros can play a role there. And I just would like to finish that with the fact that although changes in cardiac function occur quite late, only after 12 months in the MDX mice, the structural changes are evident very early. So even in 3 months old mice, as well as very important LV fibrosis that is present in these mice. So we now want to investigate whether Ros are important also there. So I would just like to finish with the slide that I have been showing you at the beginning. So in conditions in which there is a spillover of catecholamines or neurotransmitters into the plasma, and these are heart failure, for instance, heart failure patients have a spillover of norepinephrine into the plasma. This happens also in muscular dystrophy patients. So this mechanism seems to be important from this mechanistic point of view. So this
4 minutes left.
Okay, I'm finishing. This spillover of catecholamines is due to reduced uptake of the catecholamines back into the sympathetic nerve efferents. And so this allows more a neurotransmitter to enter the extraneuronal cell. In fact, in heart failure patients, it has been shown that extraneuronal monoamine uptake is doubled. In fact, you have higher influx of catecholamines into the cytosol of extraneuronal cells, higher MAO activation and H2O2 formation that can target mitochondria to further exacerbate this oxidative stress. On one hand, they can also, I didn't show you this, they can target metalloproteinases and mast cells to induce degranulation and extracellular matrix remodeling and fibrosis. They can also target other neighboring organelles such as ER to induce ER stress, impairment in calcium homeostasis and contractile protein oxidation. And they can also target mitochondria to induce opening of the PTP, release of cytochrome C, caspase activation and apoptosis. So I just would like to finish by thanking the people that have contributed to these projects, especially Fabio Di Lisa, with whom I have been working for many years. And thank you for your attention.
Thank you. Question, please. I have, as usually, a general question. How much of your wonderful data are relevant to skeletal muscle fibers? And it could be that they are more relevant to the slow type, which is less distant than fast fibers from the cardiomyocyte. Is this a crazy idea, as usual, of Hugo, or there are some rationale?
No, you're right. It's most likely, I mean, it's probable that slow fibers are more susceptible to this mechanism, but I think that the data that I have shown in muscular dystrophy animal model, but we have done this also in myoblasts from patients from patients with collagen 6 myopathies. We think that this mechanism is quite universal. So by preventing this Ros formation, we can preserve organelle dysfunction, not only mitochondria, but also from neighboring organelles. So I think that the data I have shown also relative to muscle dystrophy is quite encouraging in that sense, saying that this is a universal mechanism that can be, let's say, you can take advantage of it in different pathologies, also those including skeletal muscle.
Corrado, Angelini, comment?
Yes, I have a question. I appreciate very much your talk, and you have two models that you quoted of muscular dystrophy. One is the well-known collagen 6, that's somewhat similar to the I mean, what we call Bethlehem myopathy or other variety. The other one is MDX, that is similar to Duchenne, you know.
Yes.
And while it is well known that in Duchenne dystrophy there is cardiomyopathy, and even in Becker dystrophy, what about the Bethlehem? Do you have any cardiomyopathy there?
We haven't, as far as I recall, we have not looked at that. So far,
The mouse, does it have any cardiac signs?
No, I don't. Well, also in the MDX, I mean, it's a quite stronger, much stronger phenotype than the Bethlehem one, but cardiac changes are being observed only in very old mice. So this is one of the issues with animal models. And as far as I recall, no, there is no data reporting any changes in cardiac function or structure in the Bethlehem model in the collagen 6.
Yeah, that's as a clinician, I've never heard that there were cardiac signs in the Bethlehem. That's why I asked you.
Okay.
So your monoamino oxidase dysfunction applies to what? To sympathetic dysfunction in the heart of Duchenne, Becker. How do you apply this to humans?
Well, first of all, we have to see whether this is the case in humans. So this is quite not under investigated field. So nobody has ever looked at that, and we are starting to look at that now. Duchenne muscular dystrophy is also associated with the activation of the sympathetic nervous system and elimination. For instance, catecholamine metabolites have been found in urine of these patients. So I think this mechanism could be relevant in the Duchenne muscular dystrophy model and can affect also the heart, but we still have to prove that.